Pharmaceutical Chemistry-I. Limit test. Quality of medicinal agents should be of high standards as these are administered to the patient suffering from some diseases. Medicines should not cause adverse effects due to impurities. Presence of impurities may also produce undesirable changes, incompatibilities, during dispensing or processing in the laboratory/industry. The Pharmacopoeia is the official compilation of standards of quality for all currently used medicinal agents together with methods used for such quality control. With the use of modern techniques, chemical/medicinal agent can be obtained possessing high states of purity. In some cases, such purity is achieved at relatively reasonable cost. In many cases, however, the purification cost may be quite high. To keep the cost of medicament at a low level, some compromise has to be made in respect of impurities without ignoring the ethical aspect as well as patient's health aspect. The impurities should not be in a quantity that may harm the patient or the processes of manufacture, storage, stability, etc. Hence, Pharmacopoeia has fixed the limit of various impurities, based on toxic effect of particular impurity. If the impurity is present above the limit, disastrous consequences may follow. To check this limit test is carried out. Impurities are introduced in medicinal agent from the raw materials and process/equipment used for its manufacture or preparation. Limit tests are conducted for checking the limit of chloride, sulfate, iron, arsenic and heavy metals. Limit test for chloride. It depends on production of white turbidity/opalescence on addition of silver nitrate solution to sample solution in presence of nitric acid. C plus agno, AgCl plus NO3, white opalescence. The intensity of white opalescence is compared with standard opalescence containing known amount of chloride. Less intensity in sample solution is comparison to standard solution indicate the presence of chloride impurity within limit. Limit test for sulfate. It depends on production of while opalescence on addition of barium chloride solution to sample solution containing hydrochloric acid. So plus BaCl2 BaSO plus CH, white opalescence. Limit test for iron. It depends on production of pink to violet color on addition of thioglycolic acid to sample solution in alkaline solution. Fet plus 2HS.C.H. 2CuFas. CH. COO2 thioglycolic acid ferrous thioglycolate, pink to violet color. The intensity of color is compared with the standard color, developed from known amount of iron solution. Limit test for arsenic. It is based on production of yellow stain on mercuric chloride paper, formed by reaction with arsine, ash, gas. This gas is formed by reaction of sample solution, containing arsenic, with nascent hydrogen. H1SO arsenic acid or HSO, plus 3H2 ash, T plus 3H2O. Arsenious acid arsenigous ash HgCl ash, HgCl2. Yellow colored complex. The intensity of color is compared with a standard color formed from standard solution containing known amount of arsenic. Limit test for lead. It depends on production of opalescence on addition of sodium sulfide solution. Hydrogen sulfide is added in case of heavy metals. Pb++ Na2S2 Na++ Pbs. The intensity of sample and standard solution are compared. Gastrointestinal agents. These agents specifically act on gastrointestinal tract. Some of these inorganic agents include acidifiers, used in the treatment of achlorhydria, antacid, used in the treatment of hyperacidity, purgatives, used in the treatment of constipation, antidiarrheal, used in the treatment of diarrhea, etc. Gastric hydrochloric acid acts by destroying the bacteria in ingested food and drinks. It softens the fibrous food and promotes the formation of the proteolytic enzyme, pepsin. This enzyme is formed from pepsinogen at the acidic pH, below 6. Pepsin is helpful in the metabolism of protein. Hence, lack of hydrochloric acid can cause gastrointestinal disturbances. Achlorhydria is a disease where there is lack of hydrochloric acid in gastric secretion. Dilute hydrochloric acid is administered in the treatment of achlorhydria. Antacid are alkaline bases used to neutralize the excess of gastric hydrochloric acid associated with gastritis and gastric ulcers. The beneficial effect of antacid are attributed to two primary mechanisms. I. Neutralization of gastric acid. 2. Reduction in the proteolytic activity of pepsin. Antacids are classified as follows. I. Systemic, absorbable, for example, sodium bicarbonate. They are soluble, readily absorbed and capable of producing systemic disturbances and symptoms of alkalosis. 2. Non-systemic, non-absorbable, for example, calcium carbonate and basic aluminium compounds. They are not absorbed and act exclusively in gastrointestinal tract, and their adverse effects are generally limited to changes in bowel function, that is diarrhea or constipation. Cathartics, purgative, affect fecal consistency, accelerate the passage of feces through the colon and facilitate elimination of feces from the rectum. Laxatives are mild cathartics. Consequently, they are used to ease the pain of elimination of feces in anorectal disorders such as hemorrhoids and fissures. These can be conveniently classified into 5 groups. I. Stimulant laxatives. Phenolphthalein, castor oil. 2. Saline laxatives, magnesium sulfate, potassium sodium tartrate. 3. Bulk forming laxatives, natural and semi-synthetic polysaccharides. 4. Lubricant laxatives, mineral oil. V. Fecal softener, docusate sodium. Adsorbent, chemically inert powders, are generally used in antidiarrheal formulation. These have the ability to absorb gases, toxins and bacteria. The fine state of subdivision of these inert powders confers high absorptive capacity upon them. Light kaolin, hydrated aluminium silicate, is generally used to absorb bacterial toxins in diarrhea and chronic ulcerative colitis. Electrolytes. All the living cells contain fluid and electrolyte. The intracellular fluid, present inside the cell, is twice in volume to extracellular fluid, present outside the cell. The normal physiological function are very much dependent on proper maintenance of body fluids and different electrolytes in extracellular and intracellular fluids. Physiological functions of various cations and anions are as follows. Physiological function of cation. Sodium I. In association with chloride and bicarbonate, it is required in regulating the acid-base balance. 2. It maintains the osmotic pressure of various body fluid compartment. 3. It is essential for the normal irritability of muscle and permeability of cell. Potassium I. It is essential for muscle activity, especially of cardiac muscle. 2. It controls the acid-base balance, osmotic pressure and water retention. Magnesium I. It affects the neuromuscular irritability. 2. It is an activator of some enzymes involved in carbohydrate metabolism, for example, hexokinase, phosphorylase, phosphoglucomatase. Physiological function of anions. Chloride I. It is required for maintenance of proper hydration. 2. It is essential in regulating the osmotic pressure and acid-base balance in the body. 3. It is essential in the formation of gastric hydrochloric acid. Bicarbonate I. It maintains the acid-base balance and hence, act as buffer. 2. It also regulates the CO2 transport, proper hydration, gastric acid and electrolyte balance. Phosphate I. The combination of monohydrogen phosphate and dihydrogen phosphate act as buffer system in body. 2. It is a constituent of bones, teeth, nucleoprotein, phospholipids, etc. Sulfate. It is a constituent of protein mucopolysaccharide, heparin, thymine, etc. Mineral nutrients. Human body requires a number of inorganic cations, ferrous, calcium, zinc, manganese, copper, and anions, iodide, fluoride, etc., for proper functioning. Nutritional deficiencies as well as some conditions of ill health, diarrhea, vomiting, surgery and side effect of drug may lead to inadequate supply of such essential ions. Hence, these must be supplemented with mineral nutrients. Some diseases which occur due to nutritional deficiencies are as follows. Tetanus. It occurs when there is a lack of calcium ions in plasma. These are tingling sensations in the limb, and increased neuromuscular irritability with twitching in the skeletal muscle, laryngeal spasm and finally convulsion. Rickets. Severe calcium deficiency causes rickets, a disorder characterized by inadequate calcification of the bones which remain soft and become curved outward. Osteomalacia. It is a progressive weakening of skeleton occurs in adults due to calcium deficiency. Hypochromic microcytic anemia. It is a result of iron deficiency. Depending on the severity, the anemia is accompanied by tiredness, palpitation, sore tongue, etc. Cretinism. It occurs due to deficiency of iodine, characterized by feeble-mindedness and deaf mutism. Goiter. It is characterized by enlargement of thyroid gland, occur due to deficiency of iodine. Expectorants. Emetics and antidotes. Expectorants are helpful in eliminating bronchial secretions by inducing cough. They may cause this by irritating the gastric mucosa. The inorganic expectorant are ammonium chloride, potassium iodide, sodium iodide and related substances. These are classified according to their action, directly or reflexly. Volatile oils. For example, eucalyptus oil when administered orally by steam inhalation increase the respiratory secretion. They are direct expectorant. 2. Fluids liquefy the mucus and aid its expulsion. 3. Ammonium chloride and ammonium bicarbonate are reflex saline expectorants. These agents irritate the gastric mucosa and stimulate respiratory tract secretion. 4. Alkaloid, Vasicine, isolated from Adhetoda vasanja leaves, is expectorant due to its bronchodilation action. Emetics are compounds which cause emesis to expel out the contents of the stomach. They may act by stimulation of the chemoreceptor trigger zone, for example, morphine, or act peripherally e.g. antimony potassium tartrate, ammonium chloride. Emetics are useful in the treatment of poisoning and in low devices. It may be added in cough preparation. Mild emetic response stimulates flow of respiratory tract secretion. Ammonium chloride mostly and antimony potassium tartrate to a limited extent are added in cough preparation. Antidotes react specifically with an ingested poison or toxic substances or an overdose of a potent drug. They either neutralize the poison or its toxic effect, pharmacologically, antagonistic, or chemically by converting them to non-toxic or less toxic forms, for example chelates, acid, insoluble derivatives. Sodium thiosulfate act as chemical antidote for the poison, cyanide. It converts the cyanide into non-toxic thiocyanate. Sodium nithoate act as physiological antidote. It converts hemoglobin into methemoglobin in order to bind cyanide. Inhalants. Several inorganic gaseous substances are used as medicinal agent. As they are administered through respiratory system, they are called inhalants. Topical agents. Topical drugs are the compounds which are applied to the skin and mucous membrane for their local effects. Sometimes they penetrate into the deeper tissues, absorb into the blood. For example, antiseptic penetrate into the tissues to help in healing of deep infection. Topical agents are also applied within body cavities that open outside, oral, vaginal and colonic cavities, to exert its local action. Topical agents are broadly classified as follows. 1. Protective and adsorbents, talc, bentonite, zinc oxide. 2. Antimicrobial, potassium permanganate, iodine, boric acid, povidone. 3. Astringent, alum and zinc sulfate. 4. Demulcents, benzoin. 5. Emollients, castor oil, coconut oil. 6. Cleansing agent, hence aluminium chloride, sodium lauryl sulfate. Protectives. These are the inert substances applied to the skin to cover and protect epithelial surfaces, ulcer and wounds or prevent the irritation due to rubbing or friction. Protectives are generally applied as dusting powders, suspension or ointments. The compound used as protectives should have the following ideal properties. Insolubility. Protectives due to insolubility are not absorbed through the skin, hence, they are not easily washed out. 2. Inertness. They should be biologically inactive to avoid interaction with tissue. 3. Particle size. These agents are usually applied to the areas of the skin which are under constant irritation due to moisture or friction. Protectives must be subdivided to provide larger surface area, so that particle can adhere to each other, adhere better to the surface of the skin for quick moisture absorption. A fine particle also offers a smooth surface which helps in preventing irritation due to rubbing or friction. Astringents. Astringents are the compounds which precipitate the proteins. A salt of aluminium and zinc commonly used topical astringents. They have following actions. A. Styptic. These agents prevent bleeding from minor cuts by enhancing blood coagulation and constriction of small capillaries. B. Antiperspirant. These agents decrease the perspiration rate due to constriction of pores at the skin surface. C. Miscellaneous. Astringents reduce inflammation by restricting the supply of blood to the surface of mucous membrane. Astringent in higher concentration can also remove the unwanted tissue on the skin. Antimicrobials. The term antimicrobials are the compound which act against the microbes. It includes antiseptic, germicide, disinfectant. Antiseptic. This is a chemical used to prevent sepsis, but the term is generally used for a substance applied to the tissues of humans or animals to inhibit or destroy invading organisms. Germicide. It is a chemical substance used to kill bacteria. The corresponding terms of viruses and fungi are viricide and fungicide respectively. Disinfectant. It is a chemical used to destroy the microbes that cause disease in man. Animals or plants. The term is commonly applied when destruction takes place in or on inanimate objects. Mechanisms of action. The inorganic antimicrobial act by oxidation, halogenation and protein precipitation. Oxidation. Certain inorganic compounds like hydrogen peroxide, permanganate, halogen, chlorine, iodine, and oxohalogen anions oxidize the reducing group present in the protein. The commonly occurring reducing group is sulfhydryl, SH, present in cysteine. Protein performs specific function in the microorganism, and reduction process alter the conformation, shape, of the protein and hence alter its function. It results in destruction of microorganism. Halogenation. Hypochlorite, OCI, type of antiseptic or disinfectant react with amino groups present in the protein molecule, chlorination, and alter the function of specific protein due to substitution with chlorine atom for the hydrogen. Produces changes in forces, hydrogen bonding, responsible for proper conformation of protein. Protein precipitation. Metallic ions like silver, mercury interact with proteins to show antimicrobial action. This interaction may be non-specific or specific. The interaction with protein is non-specific, and at particular concentration, it kills the microbes due to protein coagulation. Some metals, e. mercury, arsenic and antimony, form strong covalent bonds with specific enzyme protein systems, particularly having sulfhydryl, SH, group to kill the microbes. Dental products. These include mouthwashes, oral hygienic product, dentifrices, abrasives, varnishes, cavity liners, descasitizers, alveolar analgesics, irrigation solution, etc. Abrasives. These are preparations used by the dentist in cleaning and polishing teeth. They are not prescribed for patients' self-use as they may be too harsh for continued or unsupervised use. Calcium carbonate in very fine powder, in form of paste or powder with other ingredients like coloring solution, etc., is used as abrasive. Generally, powders are more effective. Dentifrices. It is a substance applied with toothbrush for cleaning the accessible surfaces of the teeth. Commercial dentifrices are available in the form of pastes and powders. The powders and pastes contain abrasives such as calcium carbonate, calcium phosphate, magnesium carbonates, and phosphate. Toothpaste contain liquids such as glycerin, propylene glycol, sorbitol, and thickeners such as starch tragacanth. Dentifrices usually contain non-carbohydrate sweetening agent, but a few contain sugar. It is seen that individuals vary considerably in their need for an abrasive in dentifrices. Generally, dentrific powders are more abrasive than pastes. Some individuals who require only a slight degree of abrasion to keep away the teeth from staining may find that a mixture of baking soda and sodium chloride is satisfactory. The fluoride, like sodium fluoride, is often added in toothpaste used as dentifrice, earrest, varnishes, cavity liners. These, also called cavity liners, are used to seal dental tubuli in deep-seated cavities to protect the pulp from acid-containing dental cement. Cavity lining preparations, which depend solely upon the development of a film intended to be impervious to aqueous acid, are believed to be inadequate because of the difficulty in maintaining the integrity of film under filling condition. Several commercial preparations are available which utilize the chemical effect of calcium hydroxide for neutralizing the acid derived from acid dental cement. These contain various % of calcium hydroxide suspended in aqueous methylcellulose solution.